All right, so this is Unit 3, Metabolism, and we're going to start this unit out with a lecture on the overview of carbohydrate metabolism. And that's really an overview of all of metabolism, because metabolism in the human body really revolves around carbohydrate metabolism. And it's really a 30,000-foot view of carbohydrate metabolism to just give you an idea of the general processes and how they interrelate, so that it makes more sense as we go through those individual processes in the subsequent lectures. So first, the nuts and bolts of carbohydrates. So carbohydrates, also known as sugars, are the primary source of metabolic energy in humans. Monosaccharides are the most basic form of carbohydrates, and that's because they cannot be further broken down into another compound. Chemical formula is CH2O, X, where X is at least 3, so you can't have this be a 1 or a 2. And then they're classified by the number of carbon atoms their structure has. So if you remember these structures from your organic chemistry days, each of these angles equals a carbon. So this would be 1, 2, 3. So this is a triose. Tetrose, 1, 2, 3, 4 carbons. So that's a tetrose. A pentose has 5 carbons. Hexose has 6 carbons, and so on. So glucose, which is the major source of energy for cells in the human body, is a hexose. And you can see that structure here, 6 carbons. And it's actually the only source of energy for the brain in any state other than prolonged starvation. And we'll go through that in subsequent lectures. Other major monosaccharides include fructose, which is converted to intermediates in the glycolytic pathway. Again, we'll devote an entire lecture to the glycolytic pathway. You'll see where that plays in. And then you have galactose and ribose. And ribose is actually a component of RNA and then a precursor of DNA as well. So you have disaccharides, which are essentially 2 monosaccharides bound together. And these are commonly parts of foods that we eat. And so this is really where metabolism begins, because it begins with us eating food. And the way we get carbohydrates into our body, the form they are ingested in, is as typically as these disaccharides. And then these disaccharides are subsequently broken down by digestive enzymes in the GI tract, and then they're absorbed individually as monosaccharides. So a few common examples here. You have sucrose, which is glucose bound to fructose. So this gets ingested, you break this down. This is glucose, this is fructose. And so glucose is absorbed as a monosaccharide, and then fructose is absorbed as a monosaccharide. It's not possible to absorb this as a disaccharide. Same thing down here, lactose, which is typically found in milk and dairy products. It's a glucose bound to a galactose, broken down. Glucose is absorbed, galactose is absorbed. This is where lactose intolerance stems from. The lactase enzyme, which breaks down lactose, is either deficient in certain individuals or not as active, and so they're not able to digest lactose as properly, and so that's where that stems from. And then lastly, you have maltose, which is basically 2 glucose molecules stuck together, so you break it down. This is a glucose, this is a glucose, and then they're absorbed individually. And then polysaccharides, these are more than 2 monosaccharides bound together, 3 or more, and they're commonly function as glucose storage mechanisms such as glycogen. And we're going to dedicate an entire lecture to glycogen metabolism. So glycogen is a highly branched structure, and it's essentially a bunch of glucose monosaccharides bound together like this. So each of these are glucoses, and they're bound together in a highly branched structure, and the main function of glycogen is for storage, and it's storing glucose for essentially kind of a rainy day, essentially where you're fasting for a long period of time, or during, such as during exercise when you have high energy states as well that demand a lot of energy. All right, so now that we've covered the basics of carbohydrates, now we're going to take a step and look at this diagram here that's in your book, and this is really going to give you a 30,000-foot view of both carbohydrate metabolism and really metabolism in general. And the reason we're going to go through this is because we want to show the relationships between these different processes, because a lot of times when you learn biochemistry the first time, you learn about each of these processes individually, and then you hear about how glycolysis is related to fatty acid metabolism, or how amino acid metabolism is related to the citric acid cycle, and you haven't really been able to draw how those all connect. And so this is to give you kind of a 30,000-foot view of how all these processes are generally related. Now, we're going to have a series of lectures in this unit that will go through each of these processes individually, go through the details. This is really just to give you an overview and give you a general sense of how metabolism overall works together to produce energy for cells and for the body in general. So, carbohydrate metabolism revolves around the breakdown of glucose to generate ATP. It's all about generating ATP. And so all ingested starches and other sugars such as lactose and sucrose are converted to glucose so that then it can undergo glycolysis to start out metabolism. So glycolysis, again, we're going to have an entire lecture devoted to glycolysis, the steps, the regulation. But in general, it's a pathway that occurs in the cytosol of every cell, and it consists of a series of steps that break down glucose to pyruvate. Now, when you get down to pyruvate, there's one of two pathways that can result. You can either have anaerobic respiration, where you can convert into lactate, or you can have aerobic respiration, where you convert it into acetyl-CoA and enter the citric acid cycle. Regardless, this process of glucose being converted to pyruvate generates 2 ATP. So glycolysis always produces 2 ATP molecules at the minimum. Like we said, in anaerobic respiration, pyruvate is converted to lactate, which is a waste product and does not utilize oxygen. Now you may ask, why would you anaerobic versus aerobic? Could be a number of things. Since this doesn't use oxygen in such as states of hypoxia, when you have a deficiency of oxygen, it may be forced to go into the anaerobic pathway. Some cells, such as red blood cells, don't have mitochondria, so you can't carry out aerobic respiration. And so in those cells, it's always anaerobic respiration. So for aerobic respiration, you have pyruvate, which is then crosses from the cytosol into the mitochondria and is converted to acetyl-CoA. Now in this diagram, we have acetyl-CoA out here because it's such a central player in a lot of different pathways. But in reality, pyruvate comes into the mitochondria, converted to acetyl-CoA, and then it enters the citric acid cycle. Now the citric acid cycle generates 2 molecules known as NADH and then FADH2. And these are electron carriers that transport electrons to the electron transport chain, which is a series of membrane transporters in the inner membrane of the mitochondria. And they generate this hydrogen gradient, and we'll go through these details in much more depth in the next lecture. But anyways, that hydrogen gradient powers a protein called ATP synthase, which synthesizes ATP using oxygen. And so overall you have glucose, which is metabolized to pyruvate. Pyruvate enters the mitochondria, converted to acetyl-CoA, goes into the citric acid cycle. You generate these electron carriers, which then undergo a series of oxidation reduction reactions in the electron transport chain, which then result in generating ATP using oxygen. Now, the electron transport chain way, using ATP synthase, generates a significantly higher number of ATP compared to anaerobic respiration, which only generates 2. So in terms of generating ATP and in energy production, aerobic respiration is much more favored. Now again, there may be circumstances such as hypoxia, or a cell doesn't have mitochondria, where you can't undergo aerobic respiration and you're forced into anaerobic respiration. Acetyl-CoA is a major player because it can be used to generate a number of things such as fatty acids. As you can see here, we have adipose tissue shown here. So you can generate fatty acids, which then can combine with glycerol to form triglycerides, which are stored in adipose tissue. And this is interesting because, overeating and an abundance of glucose can eventually contribute to weight gain. And the way that happens is first, you're going to store glucose as glycogen, but that store fills up. And so if you have maxed out your glycogen and you keep producing acetyl-CoA, eventually you have produced enough energy, and then that acetyl-CoA will get shunted essentially over to fatty acid production. And then those fatty acids will combine with glycerol and be stored as triglycerides in adipose tissue. And that's how overconsumption of carbohydrates can contribute to weight gain in the human body. Acetyl-CoA can also be used to produce ketone bodies, which are produced in the liver, and they're used as a primary energy source by the brain during significant prolonged starvation. And we'll get into that in a little bit here. Acetyl-CoA can also be used to produce cholesterol. Now, kind of just again, a very general overview of how these all work together during periods of fasting and starvation. Glucose is low and then glycogen is depleted. So you have low glucose, you've used up all your glycogen storage. So at this point, you need to get energy from other sources. And those main 2 other sources are triglycerides and proteins. Now first, triglycerides. You break it down to fatty acids and glycerol. Glycerol can then be used in the glycolytic pathway, and mainly it's going to be used in this process known as gluconeogenesis. Gluconeogenesis is a process that primarily occurs in the liver, and it converts pyruvate into glucose, which that glucose can then be used by other tissues for energy metabolism. Fatty acids then undergo fatty acid oxidation or breakdown within the mitochondria. That's what this represents here, to then produce acetyl-CoA. This acetyl-CoA produced by fatty acid breakdown can then enter the citric acid cycle, which then again produces ATP. So as you can see, breakdown of fatty acid contributes to aerobic respiration and significant ATP production. So proteins, you don't like to break these down as much because they're such a critical component of so many different structures in the body. But if it gets to that point of significant fasting, proteins can be broken down into amino acids. Amino acids themselves can be metabolized to produce acetyl-CoA, which then again can enter the citric acid cycle, electron transport chain, and produce ATP. And that produces a byproduct of ammonia, which is then used in urea production, which is a waste product. Amino acids themselves can also be used as citric acid cycle intermediates, where they enter the citric acid cycle at different points throughout the cycle, and then again contributes to production of those electron carriers NADH and FADH2. Lastly, prolonged starvation shifts acetyl-CoA toward ketone body production. And so you start producing ketone bodies because the brain primarily relies on glucose. And during prolonged periods of starvation, glucose is severely depleted and not in very short supply. And so you end up producing ketone bodies, which can then be primarily used by the brain. And then that again can help preserve any kind of glucose stores and other sources of energy for other parts of the body. All right, and lastly here, this is a table. It's in your book, and it's just meant as kind of a summary of what I just said. It gives a very brief summary and purpose for each of these metabolic processes. And again, each of these processes is really going to have its own lecture dedicated to it in this unit. And just again, glycolysis, it's a pathway. It's in the cytosol. That's important. Glucose to pyruvate. You can either have aerobic respiration or lactate, which is anaerobic respiration. Glycogenesis is a series of reactions that results in the synthesis of glycogen. This occurs in the cytosol of the liver and in the muscle. Glycogenolysis is the breakdown of glycogen stores in the liver and muscle yield glucose during periods of fasting. Citric acid cycle, this is a series of reactions that convert acetyl-CoA to CO2 and water, while also generating a proton gradient in the mitochondria that drives ATP formation, like we said. Electron transport chain, these are kind of in tandem, part of the same lecture. This is a series of protein complexes in the inner mitochondrial membrane that couple transport of electrons with ATP formation. That's the key concept. Couples transport of those electrons, and the way it does that is by generating a hydrogen gradient that I was talking about that then drives ATP formation. Gluconeogenesis, this is synthesis of glucose during periods of fasting within the liver and the kidney cortex, and this provides glucose for energy production in peripheral tissues. Lastly, the pentose phosphate pathway. Didn't really talk about this much. This is one of the lower yield pathways, but again, we have a nice small lecture dedicated to this as well. This is a pathway that oxidases glucose 6-phosphate to generate NADPH and pentose phosphate. Really important in red blood cells, and again, we'll go over that in that respective lecture. All right, so we'll finish out this lecture by talking about the metabolic capacity of tissues. This is a major concept to understand for metabolism. And it's really important to understand how the metabolic capacity of certain tissues relates to their function and relates to their role in metabolism, and then also in just general physiology as well. So the liver is a major organ for biosynthesis. The livers could perhaps be the most important organ for metabolism. And it's very rich in mitochondria, and that's really important because by having a high number of mitochondria, it gives you a high capacity for oxidative respiration. So the liver is a site of high levels of oxidative respiration or aerobic respiration. So going this route versus to lactate. The liver and the kidney cortex are the only tissues that can synthesize glucose via gluconeogenesis. So that's where you take it from precursors such as pyruvate or other molecules, and you produce glucose. And in the liver, glucose is actually produced to be used for peripheral tissues. So I think we say that here. Liver synthesizes glucose during fasting from a wide range of compounds such as pyruvate, lactate, amino acids, glycerol, and fructose. The liver can also synthesize fatty acids from glucose and make cholesterol from acetyl-CoA. And you can see that here, where you have acetyl-CoA, and this is where you've maxed out the glycogen stores, you have enough energy produced through aerobic respiration. You're going to store that extra by converting it into fatty acids, which then are converted into triglycerides. The other thing about liver is it's the only tissue in the human body that can synthesize urea via the urea cycle. So you remember here, amino acid metabolism produces ammonia, which is then used in the liver as part of the urea synthesis process. which is then excreted in the urine as a waste product. The biochemical processes within skeletal muscle tissue serve to provide ATP for muscular contraction, which, as you can guess, has a very high energy demand. Muscle is composed of the following two tissue types: white fibers and red fibers. White fibers you want to think of almost for sprinting, and red fibers for marathon or long running. Now, obviously, it can be for other activities as well, but I think it's just an easy comparison to help keep them straight. So white fibers are characterized by fast contraction, but they're easily fatigued. So very quick, like in sprinting, using energy very rapidly. And so it primarily relies on anaerobic respiration because it's much quicker. You break down glucose through anaerobic respiration via glycolysis and glycogen storage breakdown, and you get ATP very quickly. However, because of that, you can blow through your stores very quickly, and that's why you can become easily fatigued. Versus red fibers are kind of more slow and steady, slower contraction, able to work for longer periods of time, and they contain high amounts of mitochondria and rely on aerobic respiration as a result of that. So you can see the mitochondria content really is a major factor for the metabolic capacity of a tissue. The other thing is red fibers can utilize fatty acids and ketone bodies in addition to glucose. The other thing is muscle is the site of the majority of protein in the body. So during starvation, protein in muscle can be broken down into amino acids that are further metabolized by cells via either being converted into acetyl-CoA, as we show here, which then can enter the citric acid cycle, or amino acids can then enter as intermediates into the citric acid cycle, which both end up producing those electron carriers, NADH and FADH2, for the electron transport chain, producing large numbers of ATP via aerobic respiration. Cardiac muscle is very rich in mitochondria. That's because it's continuously working throughout the life, literally every second of your life, and it provides, as a result of that, a high capacity for oxidative respiration. And so cardiac muscle relies primarily on aerobic respiration, producing high amounts of ATP. The other thing is that cardiac muscle has a limited capacity to break down protein and amino acids. And that makes sense. You don't want to be breaking down heart muscle tissue or heart muscle structure because that's just damaging to the heart and counterintuitive. And so that makes sense that it has a limited capacity to get energy via amino acids. Adipose tissue is the primary site for storage and mobilization of triglycerides. So you have adipose tissue over here. As far as overconsumption of glucose, again, you max out your glycogen. Pyruvate gets converted to acetyl-CoA, which can then be converted to fatty acids, combine with glycerol, and become triglycerides. On the flip side, if you're fasting for a prolonged period of time, you can mobilize triglycerides, which then are converted to fatty acids, and then fatty acids are broken down into acetyl-CoA, which then can enter the citric acid cycle to generate a large number of ATP. This fasting is where you hear about people that will fast to lose weight. What they're doing is, obviously decreasing their glucose consumption and then making their body switch over and rely on breaking down triglycerides to provide energy for metabolism. And so as a result of that, you break down triglycerides, and the thought is you end up decreasing your adipose tissue content, and then hopefully for that individual, they end up losing weight. So the brain requires a high amount of energy for ion pumping, which is then required to generate electric currents in neurons. And that's how the brain functions and how it generates thoughts and memories and other actions. And so glucose is the primary metabolite, mainly because the blood-brain barrier does not allow the entry of fatty acids nor amino acids. Not ketone bodies. Apologize, that's an error in the book. Make sure you cross out ketone bodies. Ketone bodies actually can cross the blood-brain barrier, which is important during prolonged starvation, which we'll talk about in a second. So it blocks amino acids. So as a result, the brain has a very high capacity for oxidative respiration because you need high levels of ATP, to help fuel this ion pumping, to help the neurons carry out their function of generating electric currents. So as a result of that, the brain is not able to survive for very long on low oxygen or in hypoxic states. So that's why significant hypoxia or prolonged hypoxia can have devastating effects on brain tissue for this reason, because if you're not providing oxygen, you can't provide ATP. Can't provide ATP, can't power the brain. The brain can use ketone bodies, which are synthesized in the liver from fatty acids, when they've reached sufficient levels in the blood during significant starvation periods. So what happens is that significant starvation, you've blown through all your glycogen, you're mobilizing fatty acids down to acetyl-CoA, and then you've generated enough acetyl-CoA where it's switched over to ketone body production, and then this gets high enough in the blood to where it can cross over into brain tissue and help power the brain, which the thought there is it helps spare what glucose is left to help power other tissues. Kidney cortex has a high rate of active transport to help drive the osmotic and reabsorptive functions of the nephron. And so in the kidney, there's a lot of these transport processes that rely both on directly on ATP and then indirectly on ATP, because the sodium-potassium pump helps establish the gradient across the cells. And the sodium-potassium pump, as we've discussed in the previous units, relies on ATP as well. And so kidney tissue needs a significant amount of ATP and energy production to help carry out its function. So it has a high metabolic capacity, especially for aerobic respiration. The other thing the kidney does is it helps control the acidity of the urine by releasing ammonia derived from glutamine, which is mobilized from muscle to the kidney. As a result of that, it's also capable of glucose synthesis as a byproduct of ammonia excretion. Because of that, renal glucose synthesis is dramatically higher during periods of metabolic acidosis, because you're trying to excrete ammonia to help control the pH within the body. And so as excreting a high level of ammonia, you can then use that to help generate glucose. Lastly, the red blood cells, they have no nuclei or mitochondria. So they have no mitochondria and they have no capacity for aerobic respiration. So they rely entirely on anaerobic glycolysis to produce ATP. So there's 2 ATP that are produced by the process of glycolysis. This is what powers red blood cells. In addition to that, glycolysis within red blood cells specifically generates a molecule called 2,3-bis phosphoglycerate, which regulates the binding of oxygen to hemoglobin. And we'll talk about that in much more detail in the heme lecture in this unit. All right, so that closes out our overview of carbohydrate metabolism. Hopefully now you're more familiar with processes involved in carbohydrate metabolism and then have a general sense of how they relate and work together. And now in the following lectures, we'll go through each of those processes individually in each lecture and talk about the significant details, and then again referencing back to the concepts and diagrams we covered in this lecture to explain how they work cohesively together.